Good afternoon. My name is Filip and I'm representing the organization ELASMOCAN, which is based in the Canary Islands, Spain. And I will introduce you to the project Angelshark-ID. Angelshark-ID is in fact a research action created for long-term monitoring of angel sharks Scotina scotina in the Canary Islands, employing photo identification. The studied species is a benthic shark with sedentary habits and a remarkable dorsoventrally flattened body, as you can see from this video. It suffered significant reductions in its historic distribution, including local extirpations, up to the level that nowadays it is presumed that the population from the Canary Islands is the only remaining stable one. However, in spite of having been a fishery resource in this region, there was a total lack of biological knowledge of it. And those parameters from other distributions were extrapolated to the local ones, resulting in erroneous species management. In addition, there are no reliable fishery records available or other historic information that could enable us to evaluate the populations. So, therefore, the aim of our study, of our project, is the long-term monitoring and generation of scientific base knowledge while minimizing research impacts. Parameters of interest include the identification and description of population structure, growth and longevity, population size, critical areas, potential threats and population indicators, but also the establishment of a tissue sample data bank for genetic and trophic ecology assessment and a motivation of citizen awareness for conservation. So, therefore, for data collection, we have conducted visual underwater sensors in 4 of the 7 islands since 2006. And upon encounters with angel sharks, we register sex and biometric measurements of the individual, physical parameters and ecological aspects of the location. We collect the tissue sample and take photographs of distinctive body marks, which are later used for individual identification. So up to now, we have characterized 19 different types of body marks, which are considered in the identification process, which enabled us to detect individuals over prolonged periods, but also juveniles during their development. So here you can see an example of the dark spot on the left pectoral fin that, together with the V-shaped nick and their relative position, allowed the identification of an individual for over 9 years. And in this example, you can see a juvenile that was observed during a period of more than 7 months and identified based on caudal fin damages of the posterior margin. So also during the 2016 sampling campaigns in Gran Canaria, we registered 173 interactions with angel sharks, of which 94% could be characterized for individual identification. 11% of these observations were resightings, which, as indicated before, provides us information about growth and longevity, and also habitat use patterns. Our technique also enabled us to detect habitats where individuals show different activities, from juvenile development to adult mating, and also to identify a micropredator and 2 parasites for the species. So perhaps these bugs could be an additional indicator for the population. Related to this, I would like to invite you to the poster session where we have some more details about this topic. And finally, our tissue sample data bank enabled us to start a study on genetic diversity and population assessment. So our results indicate that the innovative way of implementing photo identification for Scotina scotina is a viable method for monitoring based on the high percentage of individuals that can be characterized and the possibility to detect resightings on short, medium and long term. In addition, it is a technique that provides minimum perturbation to the species, while it can provide us with essential information for population evaluation, and it is therefore a potential conservation tool that meets our objective. I would like to thank the Loro Parque Fundación and Shark Foundation for funding the project, and also local and national authorities for authorizing our research activity, and yourselves for your attention. Thank you very much.